In this video, I will talk about human reproductive, oogenesis, spermatogenesis, fertilization, mitosis, and meiosis. Let's talk about the stage of mitosis. Mitosis is a type of cell division that produces two genetically identical daughter cells, and each daughter cell contains 46 chromosomes, the same number as the parent cell. Before the mitosis begins, the cell interphase. During this stage, the cell grows and the DNA in the form of chromatin and duplicate DNA. At the end of interphase, every chromosome consists of two sister chromatids joined together at the centromere. Also, the DNA has been replicated, but the chromosome number is still 46 chromosome. It's mean 2n. Next is prophase. During this stage, the chromatin condenses into visible chromosome and the nuclear envelope begin to disappear, and meanwhile the centrosome move toward opposite pole. This is centrosome and this is visible chromosome, and this is the nuclear envelope. The centrosome move toward opposite pole of the cell and start forming the mitotic spindle. This is mitotic spindle, which will later separate the chromosome. Here is separate chromosome. The cell still contains 46 chromosomes, each made of two sister chromatids. Next is prometaphase. During prometaphase, the nuclear envelope is completely breaks down, it completely disappears, and the spindle fiber, here is a spindle fiber attached to kinetochore. Which is kinetochore? Kinetochore located at the centromere of each chromosome. Here is centromere. Kinetochore is located at the centromere. Here is chromosome. Here is centromere. The kinetochore is located at the centromere. The chromosome begin moving toward the center of the cell, and the chromosome number is still 46. For metaphase, in metaphase, all chromosomes line up along the metaphase plate, which is middle of the cell. Each chromosome is attached to spindle fiber from opposite pole, ensuring that each daughter cell will receive an identical set of chromosomes. Here is daughter cell and other daughter cell, ensuring that each daughter cell will receive an identical set of chromosomes. And cell still has 46 chromosomes. Next is anaphase. the centromere split, allowing the sister chromatide to separate, and each chromatide is now considered an independent chromosome, and the chromosomes are pulled toward opposite pole. Here is pole, other pole. At this moment, the cell temporarily contains 92 chromosomes because the sister chromatids have separated. In telophase, the chromosomes reach opposite pole. They begin to decondense back into chromatin, which is mean the cell returned to initial state, and each daughter cell contains 46 chromosomes, and the new nuclear envelope is formed around each group of chromosomes, creating two new nuclear. Finally, for cytokinesis, the cytoplasm divides, producing two identical cells. Each daughter cell contains 46 chromosomes, which is mean 2n, here 2n, and here 2n, and is genetically identical to the original parent cell. Now, let's move on to meiosis. Meiosis is a special type of cell division that occurs only in the reproductive organ, which is mean the ovaries in female and the testes in male. Its main function is to produce gametes, which are eggs and sperm cells. Unlike mitosis, meiosis is reduce the chromosome number by half from 46 chromosome, which is mean to n, to 23 chromosome, which is mean n. In mitosis is still 46 chromosome. From interphase to telophase is still 46 chromosome. But in meiosis, it reduces the number of chromosomes by half from 46 chromosome to n to 23 chromosome n. This reduction is essential because when fertilization occurs, the normal chromosome number of 46 is restored. Okay? Let's start for interphase. The two, mitosis and meiosis. Before meiosis and mitosis, the cell enter interphase. During this stage, the cell grows and duplicates its DNA. Although the DNA has been replicated, the cell still contains 46 chromosomes. Okay, the chromosome number is 2n, 46 chromosome. However, each chromosome is now composite of two sister chromatids, here sister chromatide, and other sister chromatids. And meiosis is divided by two meiosis. Meiosis 1, reductional division. Reductional division is also divided by prophase 1, metaphase 1, anaphase 1, telophase 1, and the meiosis divided by meiosis 2. Meiosis 2 is equational division, and the equational division divided by prophase 2, metaphase 2, anaphase 2, telophase 2. So only mitosis is prophase, interphase, prophase, prometaphase, metaphase, anaphase, and telophase. Okay, so let's start about for prophase 1. Prophase 1 is the longest and the most important stage of meiosis. Why? Because during this stage and during this phase, homologous chromosomes pair together in a process called synapsis, forming structure called tetrad. Here, one of chromosome and other chromosome is paired together in a process called synapsis, forming structure called tetrad. Okay, and another important event is crossing over, where homologous chromosome exchange piece of DNA. This creates genetic variation among offspring, and the nuclear envelope disappear, and the spindle fiber begin to form. Okay, so begin to form. The chromosome number is still 46 chromosome, which is mean 2n. For metaphase 1, in metaphase 1, the homologous chromosome pair. So here homologous chromosome pair, here is pair, line up at the center of the cell, and each homologous chromosome is attached to spindle fiber coming from opposite pole. Here is a spindle fiber coming from opposite pole. Here is opposite pole, other opposite pole, and still the chromosome number remain 46 chromosome. For anaphase 1, during anaphase 1, the homologous chromosome, okay, homologous chromosome separated and moved toward opposite pole. But here, notice the sister chromatide remain attached. Okay, so only the homologous chromosome separated. In mitosis, the chromatide is separated, but in meiosis, the homologous chromosome separated. So here is a chromosome, okay, and here is chromatide is separated. Chromosome is separated in a chromatide, and here homologous chromosome is separated in chromosome. Okay, so and move toward opposite pole. This is what reduce the chromosome number. Okay, so here is reduce the chromosome number to 46 and 23 chromosome is later in telophase 1. In telophase 1, the chromosome arrive to opposite pole. The cytoplasm divide, here is divide, producing two daughter cell. Each daughter cell, here daughter cell and other daughter cell, contain 23 chromosome, which is mean N. But each chromosome still consists of two sister of chromatids. Okay? From meiosis to equational division. Why is called equational division? Because the chromosome number does not change. It's still 23 chromosome. Okay? Here in telophase 1, each daughter cell contains 23 chromosomes. So here transform from prophase 2. During prophase 2, the chromosome can dance again and the nuclear envelope breaks down, which is mean it disappears. If it has reformed and a new spindle fiber develop, and each cell contains 23 chromosomes. Here in meiosis 2 equational division and the same of mitosis. Okay, so the same step of mitosis for meiosis 2. It's the same steps. For metaphase 2, in metaphase 2, the chromosome lines up and really at the center of each cell, each chromosome is attached to spindle fiber from opposite pole. Here is opposite pole, and the chromosome number remain is 23. Here is a 23 chromosome and 23 chromosome. And in phase 2, during phase 2, the centromere divide. Okay, here is divide, and here is a chromatide and a chromatide. The sister chromatide separate and move toward opposite pole, and each chromatide is now considered an individual chromosome. In telophase 2, finally, the chromosome reach the pole and a new nuclear form, and the cytoplasm divide again. At the end of meiosis, 4 genetically different cells. Okay, here 1, 2, 3, 4. So 4 genetically different cells are produced. And the 4 genetically different cells, it is haploid or diploid, for sure is haploid. Okay, and each cell is contain 23 chromosome, which is mean N, with one chromatide. Here is chromatide per chromosome. Okay, so final result, 4 haploid cells are produced. Before we study oogenesis and spermatogenesis, let's first look at the human reproductive system. For human reproductive system is responsible for producing gametes, also called sex cell, and for making the hormone necessary for reproduction. The female and male reproductive system have different organs, but they work together to make reproduction possible. Let's begin with the female reproductive system. The main organ are ovary and fallopian tube, uterus, and vagina. And the most important organ for today's lesson is ovary. Why is ovary is the most important organ for today's lesson? Because has two main functions. It's produce the female gametes, so it's produce female gametes for fallopian tubes, and it's secret the female sex hormone, the estrogen and progesterone. Okay, so inside the ovary, the process called ovogenesis is take place. This is a process by which an egg cell develop and mature. Now let's look at the male reproductive system. The main organ of the male reproductive system are testes, epididymis, vas deferens, prostate gland, seminal vesicle. And the most important organ for today's lesson is testes. Why testes? Because has two main functions. It produces the male gamete called sperm cell, and it secretes the male sex hormone testosterone. Inside the testes, the process called spermatogenesis take place. This is the process of producing mature sperm cell. So for testes, produce sperm and the hormone testosterone. Epididymis store and mature sperm. Vas deferens is transport sperm. Accessory gland, prostate, add fluid. For female, the ovary is produce egg. Fallopian tube is transport the egg. Uterus is the site of implantation and development